But tonight we're going to be focusing on epigenetic orthodontics with the biomimetic DNA appliance system. And again, these words are used to describe what we are seeing. There's hard science behind the approach that we're taking. And so this is why we use these words to describe quite specifically what we're trying to do and how we're trying to achieve it. So by the end of this evening, you should know what we mean by epigenetics. Orthodontics we all know, which is straightening the teeth. And this idea of biomimetics, we're going to look at that in a bit more detail and see some cases, and then try to understand what it is that makes the DNA appliance system unique. And what we'll see here is that it's not one appliance. There are different types of appliances. And these appliances are chosen with regard to the patient's requirements. So when someone comes to the office, they may have different types of requirements. Some people have teeth which are not straight. Some people have jaws which are not well coordinated. Some people have pain such as headache or TMJ pain. Some people have issues with airways such as snoring or sleep apnea. And so the appliances, although they have the principles in common, their actual design is slightly different depending on how the patient presents for treatment. Now, what do we know about genetics? Well, the phrase is like father, like son. In other words, children tend to look like their parents to a greater or lesser extent. And we see two famous families here. Here's the Obama family, and here we see the family of the Bush family, of the Bush twins. So what we know now is that genetic inheritance has a strong influence on facial features through growth. So if the dad or the parents have got specific jaws, the chances are that the children will have a similar type of facial feature. And so because of this, we say that malocclusions, or people who don't have straight teeth, tend to run in families. An example I've given here is the Habsburg's jaw. And the Habsburgs were an Austrian-German royal family. And previously, the royals used to intermarry, and so the chances of having that gene were increased, kind of gene pooling increased. So that was the type of jaw that they tended to develop. But what I want to show you here is these two kind of modern families, and these children do look like their parents, but what we know about these two guys is that they are twins. Now, when we look at their facial features, actually they look quite dissimilar. Even though they've got so many genes in common, the outcome is slightly different. And the way to explain that is that during genetic replication, there's the idea of crossover occurring, so you get different combinations of the same genes, and what that does, it provides variation in the outcome. Just think about two dice. When you throw those two dice, there are different combinations that can be achieved, even though the two dice are the same. And so it's a little bit like that with the genes, except that there are approximately 30,000 genes in the human genome. So that gives you a very wide range of variation, from eye color, from tooth size, to all sorts of physical features that can vary from person to person within the same family. Now, what do we know about genes? Well, we know a lot more about genes now than we've ever done before in history. And so we can do molecular genetics 101. Let's just look at how these genes are organized in the human genome. So what we say is, let's look at a typical cell. So here's a cell, and here's the cell membrane around the cell. And then we find inside the cell the nucleus, and there's a nucleus in the center of the cell. And within the nucleus, what we find bundled in is the DNA, the genes. And so if we were to take that nucleus and unravel that gene, that DNA, what we'd find is that in terms of structure, it's got this sugar phosphate backbone, okay? And then along that backbone, we've got these little projections called base pairs. And these base pairs are highly specific. It's like a computer code. This will tell, it's like a programming, will say, these are the components that I have the potential to make. I may or may not make those components, but the potential is there. And what we see here are these base pairs, and what we have done very recently is decoded the entire human genome. We know that the human genome is contained on 46 chromosomes, and these chromosomes, here's a chromosome here, and the chromosome just seems like a colored body. So when the scientists first discovered chromosomes, they were kind of differently colored. So they just said a colored body, there's a chromosome right about here. But you see these bands going across the chromosome. All of these represent different genes. And so when we unravel the DNA, we see these base pairs. And what these base pairs do is they encode for a specific gene. So let's take, this gene on the chromosome right about here. From here to about there, that might be the first gene. And from here to here might be the second gene. And from here to the third gene, and so on and so forth. So all these genes are slightly different in terms of their size and their location and the things that they can do. Now, let's just look at the human race for a second. We see all these different people here. These are different ethnic groups. But what these people have in common is they have the same genes. It's a human genome, and it's identical for all humans on the face of the earth. But these guys look so different. This guy's got dark skin, these people have got light skin, and so on and so forth. What makes these people different is not that the fact that they have different genes, but they are expressing different combinations of the same genes. So it's like saying, let me go to the store and I'm going to buy some ice cream. Well, I can buy chocolate-flavored or vanilla ice cream. It's still ice cream, but it's got different flavors. And so what the human genome has is this idea of diversity. You can have the same genes there, but they're going to be expressed slightly differently. Different climate, different geography, different nutrition, all these things can affect the way those genes are being expressed and the results being produced. So what do we know about the human genome? As of today, more than 99.9% of the human genome has been sequenced. In other words, we know all the genes that are required and the base pairs for those genes that are required to make a normal, typical human being. And by doing that sequencing, we know there's about 26 to maybe 30,000 genes in the human genome. Now, what else do we know? We also know that within that human genome, certain regions are highly conserved. And so if you're going to make a tooth using the genes, then you're probably going to need a jawbone to support that tooth using different genes. And you're probably going to need a jaw muscle to move that bone for eating using different genes. So why do we just put those whole genes, that whole different groups of genes kind of together? And so what you have is morphology, shape, and information being inherited. And the genes that do that for humans or for mammals are called the homeobox genes. The homeobox genes, what they do, and a whole array of these genes, what they do is say, I'm going to make you a top jaw, I'm going to make you a bottom jaw, I'm going to make you a facial muscle, I'm going to make you a molar tooth. And so there's a lot of variation that can go on with those genes. Let's look at an example of variation. What do we see here? We see two children, two little babies, and they look very, very different. And the surprising story behind these two children is that they have the same parents. Actually, they are twins. These are babies born to an Australian couple just a few years ago, and even though they have the same genes in common, this guy is expressing genes for dark skin, and this guy is expressing genes for light skin. And it looks very unusual, and it is kind of rare, but let's give a more common example. This young guy here is the current president of the USA. This is President Obama when he was a child, and this is his late mother. And so what we see is gene segregation and expression happens all the time. We don't see it very commonly in the same generation in twins, but the possibility of having diverse gene expression exists there, and that capability resides within the human genome. Its genetic potential. So we've looked a little bit about the genes that we're going to inherit. So we inherit certain genes, we are born with certain genes, and we keep those genes for life. So what do we mean by epigenetics? Well, epigenetics is a science, and it's going to study changes that occur in the outcome of those genes. And those changes, this is changes in phenotype. So now we're looking at chocolate ice cream or vanilla-flavored ice cream. Okay, what produced that change? Well, what epigenetics says is that we're going to be able to produce changes in the outcome in the phenotype, but the underlying genes are the same. So epigenetics is a study of phenotypic changes that occur via mechanisms other than DNA sequence alteration. We're not changing the genes. It's the genes that you're born with, the genes that you inherited, the genes that you keep for life. It's your own natural genes. There's nothing artificial here. This is not like genetic modification. In genetic modification, what you do is you take a gene from a different species and artificially transplant it or transfer it to a different species. We're not doing that. We're using the genes that occur naturally and that you inherited and that you keep for life. But what epigenetics is saying is that we can produce changes in the outcome even though the genes haven't changed. So how are we going to produce those changes? Well, those changes are going to be mediated by the proteins, histones, and chromatin which surround those genes. So surrounding the genes is a region called the epigenome. And so what we have is, the genes come in a wrapper. It's just like your candy bar or your chocolate bar, it comes in a wrapper. And so this wrapper is called the epigenome. And this epigenome is capable of modification. One example I'll give you here is a process called methylation. So methylation, what's it going to do? It's going to change the wrapper in which those genes find themselves. And what that modification is going to do is it's going to allow the genes to express slightly differently, actually in a more healthy way. So what do we currently say to people, to women of childbearing age? Usually what the doctor suggests is, or the health therapist suggests, is supplementing the diet with folic acid. And what folic acid does, it produces healthier babies. And specifically what it does, it prevents neural tube defects. And how does it do that? It does that through methylation of the epigenome surrounding the genes. So actually we've been doing epigenetics for a long, long time. We just didn't realize the mechanism until more recently. So let's look at facial growth and development and find out what impact genetics has and epigenetic has on the growth and development. Now, here's the front cover of the National Geographic magazine, and this issue came out right about January of last year, about just over a year ago. On the front cover, what do we see? We see two identical twins. Well, what makes identical twins identical is the fact that they've got exactly the same genes. They've got the same genes from the father and the same genes from the mother. Genetically, they are identical. But if you look at these two young ladies here, they are slightly different. If you look very carefully, you start picking up very, very small differences, these subtle differences. And these subtle differences are epigenetic in nature. In other words, there was some epigenetic modification that occurred in one child compared to the other child, depending on the local environment of those genes and the local environment in which that child found themselves. So we can produce slight variations using an epigenetic approach. Well, if that's the case, can we take this epigenetic idea and apply it to orthodontics? Now, we know that in orthodontics that the teeth are not quite straight, the teeth are crooked. And so what we're saying is we have genetic potential to redevelop that facial region, that jaw region. I wonder if we can use it to somehow straighten out the teeth and change the bone so we get a slightly better outcome. So what do we mean by epigenetic orthodontics? Epigenetic orthodontics use the person's natural genes to correct and straighten the jaws, teeth, soft tissues, and functional spaces painlessly using biomimetic appliances. So here's a slight twist on the way that orthodontics is going to be provided. It's an epigenetic approach. We're going to take the person's natural genes, Okay, there's nothing artificial here. The genes that they were born with, and we're going to try to work with those genes to straighten the jaws and the teeth, but as well as the soft tissues and the functional spaces. So we're going to say, can we make the top jaw a little bit broader? Can we get the lower jaw to move slightly further forwards? Can we get these teeth, which are kind of irregular and crooked, can we get them to straighten out a little bit better without removing teeth because we have more bone volume to work with? And if we can get those teeth to be straightened out, can we use the tongue and the soft tissues, the lips, to keep those teeth in position once they're being corrected? And if that tongue is coming upwards and forwards and resting on the palate, well, behind that tongue is going to be a functional space, and that space is the upper airway. And so if that tongue comes forward and sits on the palate, the airway behind that tongue is going to open up and allow that patient and allow that person to breathe more freely. Now, the idea behind epigenetics, orthodontics, is to do all of that stuff completely painlessly. It's going to be done using a pain-free procedure. So why is it that we can provide a pain-free protocol in this example? Well, the answer is that we're going to use the genes that produce natural growth and development. So take a young child. The child is growing and developing normally, and as they grow, they make new bone tissue, they make new muscle tissue, and so on and so forth. Well, let's take the same process of growth and development and apply it to the facial region. Let's get that bone to regrow, and as that bone starts to improve and increase in volume, that's going to allow the teeth to line up slightly better without having to remove teeth. And because these people are growing in a physiologic way, it should be a pain-free process. So how are we going to ensure that the process is pain-free? Well, we're going to use a slightly different appliance. We're going to use a biomimetic appliance as opposed to the regular appliances that typically are used in orthodontics. So what do we mean by biomimetics? Biomimetics or biomimicry is a science. It's a science that studies natural models and uses these designs and processes to solve human problems. And so what we want to do is saying, do we have a natural model? Do we have a natural design? Do we have a natural process that we can use to solve a human problem? And that human problem might be, my teeth are not straight, I've got jaw pain, I've got headaches, or I'm snoring, or maybe I've got sleep apnea. So can we use a natural model and a design and a process to solve those types of problems? And the answer is probably yes. The natural design we're going to be using is 32 human teeth. We have a natural model for the top jaw. We have a natural model for the lower jaw, the bottom jaw, that is typically designed to accommodate 16 teeth on the top and 16 teeth on the bottom jaw. Now think about the human body. It's got a very natural design, 2 eyes, 10 fingers, 32 teeth. Well, how do you get all of those structures into the physical framework of a typical human? Well, we have to use certain processes, and the processes that we're going to be using are developmental processes, growth and development. And the amazing thing is that these developmental processes are encoded by genes. And so if we can talk to those genes, if we can communicate with the genes, we can allow those processes to be expressed, allow growth and development to go to its optimum level, and be able to accommodate the complete set of 32 teeth. And that will give you a big jaw volume, it will give you a big airway volume, and prevent any of the discomforts that we've mentioned, such as sleep apnea, crooked teeth, TMJ, and that kind of issue. Well, here's an example of biomimicry. So here's a woodpecker, and what we know about woodpeckers are very efficient at removing wood, okay? And so here's a model, a natural design, and what we've done here is taken an axe. It's been redesigned so it can be used to remove wood more efficiently. So those are the principles that we are working with when we talk about a biomimetic approach to orthodontics. That is what we're going to be doing. So here's the idea: biomimetic orthodontics, copying the natural model, mimicking the natural design. So it's still orthodontics to some extent, but it's a different approach. And so what we say is we're going to provide cosmetic treatment with health benefits. The jaws and the teeth are going to be cosmetically improved, but behind the jaws, the airway is going to be improved. That allows better sleep. If you get better sleep, you get better replenishment of the factors that you need for good brain function, for example. And so when you wake up in the morning, you'll feel refreshed. You'll have better function during the day. And long-term benefits are associated with improved sleep and improved airways. And so our idea is to provide cosmetic treatment with systemic health benefits that go hand in hand with those improvements. So what am I saying here? I'm saying that epigenetic orthodontics, it can be a very powerful technique if you can harness its potential power. So let's look at potential power. What's the genetic potential here? This is a moth. It's a Cecropia moth, okay? Well, actually, it's a caterpillar. It's a bug, okay? And what we see is a lot of segmentation, a lot of legs, and a kind of spiny type of appearance, okay? Well, this bug is going to undergo metamorphosis, and here it is here. Here's the metamorphosis, and it's going to emerge as a beautiful butterfly or a beautiful moth. And what we see here is symmetry. What we see here is coloration. What we see here is different function. And this creature here, this kind of bug that we said, this caterpillar that we saw, underwent metamorphosis to emerge as a symmetrical, beautiful-looking butterfly or moth. So what happened during metamorphosis? Because these two creatures, they have the same genes. This guy has the same genes as this guy's, but they look completely different. So what happened during metamorphosis is that different genes were expressed. It's the same genome, but different combinations of the same genes were expressed. It's like throwing the dice. You threw the dice, you got a low number. You threw the dice again, and you hit the jackpot. And so this is the analogy that we use to try to describe what we mean by epigenetics and what we mean by genetic potential. The genetic potential resides in the human genome. If we can change the conditions, we can get different genes to be expressed, we'd expect a better or a different outcome. So can we apply this example, this principle, can we apply it to our regular normal people? Well, here's a young lady here, and if you look at her facial profile, it's not too bad, except that the bottom jaw here is kind of set back a bit. If you look at her facial profile, draw a straight line, that jaw is set back. And what we've been able to do with a DNA appliance is to allow that jaw to come forward. It gives a better facial profile, and beneath that jaw profile, we get a better-looking airway, allows this young lady to be an improved facial profile with better performance in terms of sleeping and cognitive function in terms of brain function. So what makes epigenetic orthodontics different from our traditional orthodontics? And, again, let me emphasize, we work very closely with our orthodontic colleagues. This is a different way, an alternative way, but, there's no reason why your traditional orthodontics can't be done. But epigenetic orthodontics is aimed at the overall health of the facial region. And we do this by providing treatment protocols that address the underlying etiology of the underlying signs and symptoms of crooked teeth or malocclusions. So the scope here is a little bit wider. We're looking at overall health of craniofacial region. Well, the craniofacial region, that's the jaws, it's the teeth, it's the jaw joint, and it's the upper airway, and actually it also includes the brain as well. And so the craniofacial region is from the top of the head down to the bottom of the neck. And we'd like to work with the teeth, but we're looking at the overall health in a bit more, it's a wider scope. And the wider scope is by saying, let's provide a treatment protocol that addresses the underlying reasons, the causes. Why are those teeth crooked? Why do we have crooked teeth? Well, if the teeth are not straight, it means the underlying bone isn't fully developed. which means that the underlying joint, the suture which joins those bones together, hasn't been fully worked with. It hasn't been fully kind of harnessed. And so what we'd like to do is use a protocol that addresses the sutures, which are the joints between the jaw bones, and allows the bone to redevelop. If you redevelop that bone volume, then you've got more space to accommodate those teeth. It means that I don't have to take teeth out. So, we did a study to find out, how concussions are caused in athletes. And these were high school footballers, and it's a published study. And what we found is the guys who had a kind of a deep bite or had some teeth taken out, they were more likely to get concussions during sports injuries. And we also found that if we were to put a mouthpiece into those athletes, we could decrease the rate of concussion by about 80%. And so that's what we mean by craniofacial, overall health of the craniofacial region. It's not just the teeth and jaws, but by straightening and by improving that region, we have knock-on effects on things like brain health, TMJ health, and airway health. So patients with headaches or migraines or TMJ issues, they can be helped. Patients with snoring or sleep apnea, they can be helped. And so what we want to be able to do is address the overall craniofacial region by looking at the signs and the symptoms of these teeth which are not straight. And these concepts arose, by doing some basic research, clinical research on very young children, little babies. Little babies born with things like a split lip or a cleft palate, that kind of thing. Teenagers who are born with, craniofacial issues, where they had where they were born with a jaw deformity, for example, and then going on to look at adult patients, with different conditions such as sleep apnea. So let's say we're going to undertake epigenetic orthodontics. We're going to do a biomimetic correction. We're trying to copy or follow or mimic the growth and development that is encapsulated within the human genome. How are we going to do that? What are the components? Well, first of all, we'd like to correct the size and the shape of the top jaw, the maxilla. And the reason why we start with the top jaw typically is because of the way the human body develops. The human body develops from the top down. It's called a craniocaudal gradient. So if you look at an embryo or look at a fetus, we look at the pattern at which it follows, then what we find is it follows, generally speaking, this top-down gradient. So we're going to do a biomimetic approach. We're going to try to follow the body in the same way. We're going to start with the top jaw, and let's see if we can redevelop that, increase its size, change its shape. So what do you mean by increase in size? Well, we can make that jaw broader, make it wider. If it's too narrow, we can make it slightly broader. And as we are doing that, the shape of the jaw will change. Instead of having a high-vaulted V-shaped palate, we allow the palate to become broader and flatter, and that will allow the teeth to be accommodated, and it will give more space for the tongue to rest on. So let's start with the top jaw. But we're going to do something similar for the bottom jaw. We're going to correct its size and its shape and its position. So what makes the top jaw different from the bottom jaw? Well, the bottom jaw, I can quite easily move it. I can move my mandible. As I'm speaking, my bottom jaw is moving. My top jaw is relatively stable because it's attached to the rest of the cranium. And so what I can do with my bottom jaw is I can move it into position. If it's asymmetric, I can move it towards the midline. I can also change its size. I can make it broader if I want. I can change its shape by remodeling the bone. And so what we're going to be able to do here is change the foundation. This is the foundational correction. And what we want to do is the skeleton of the jaws. We want to change that, correct it, improve it. And as we do so, we can do a dental correction. We can move those teeth into better alignment because we've got better jaws to work with. But that's only part of the equation. The third part is then to re-coordinate the new top jaw and the new bottom jaw in terms of where they are in space. So this is an important point for stability, is to re-coordinate the maxillomandibular spatial relations, the positions of the top jaw and the bottom jaw in space. Is that top jaw broader? Has that bottom jaw come further forwards? And if that's the case, then we want to make sure that the occlusion, which is the way the teeth bite together, is stable. We want to make sure that the tongue is sitting on the palate because that will give you stability of the result. We want to make sure that the lips are lightly in touch when you're at rest. And if the lips are slightly touching in normal posture, then you will be breathing through the nose. So we want to encourage nasal breathing. And then what we want to do is when you're swallowing, we want the tongue to go upwards and backwards in swallowing, not downwards and forwards. And if there are any habits such as finger or thumb sucking, we want to prevent those habits as well. And what that will give you is stability of those newly achieved foundational changes. And so what we're doing in step 3 here is a functional correction. It's the way that the components are going to work. This functional correction, we call it myospatial. Myo is muscle. Muscle is mostly the tongue, but also the lips. And spatial because the teeth and jaws find themselves in a different relation in space, and it's a better relationship, so we want to be able to maintain that and keep it nice and stable throughout the life of that individual.